I do have a question. There was something in the book that just caught my attention. I'm just curious about it. So, and I figured since I'm the only one here, you have
Uh,
some time.
that's fine, Quinn. Is it something that you think other folks would be interested in?
Possibly. It was mostly in the book. I did reading last night, and steroids are a lipid. I never knew that, and I just want to know how it works. How does that work?
Oh, well, basically it works because lipids tend to have these long chains of carbon atoms. So that's kind of why they're considered to be lipids. For example, if you see page 33 of your textbook, figure 217, you see these chemical structures where you just have carbon after carbon atom after carbon atom after carbon atom, just these long chains. That's typically what's characteristic of lipids.
I did see that on the page. I mean, I remember in my chemistry class we got to draw those, and in biology too.
Yeah, absolutely. We're going to touch on some of that at the beginning of class today.
Alrighty. This should be fun. Oh. Yeah, sorry, I'm in my kitchen.
That's okay.
Because I'm closest to the basement, which has the internet thing, so. Oh, we finally get to see Nick. Hi. Anyway, I'm gonna go on mute.
Okay, good morning, everyone, and welcome back. Hope you had a nice weekend. If you can hear me okay, please type yes into the chat window. Okay, great, great. All right, so moving right along, we do have an 8-week summer semester, so things are going to start moving pretty quickly. You realize that a week from this Thursday, you're going to have your first lecture exam, okay? So let me start off today by giving you a little bit of information about how exams are going to work in this class. Okay, so I'm going to share my screen with you. Okay, just a moment. Okay. Okay, so right now, you should be seeing my desktop, and I want to show you this website that's called classmarker.com. Okay, classmarker.com is the website that each of you are going to register for with a free account in order to take exams in this course. Okay, so right now you're just seeing the opening screen for Classmarker, and just, you know, as a little bit of homework moving down the line, I'm going to have you folks register and create a free account here. Okay, so let me go back to the Word document here. I'm going to bring up the chat window so that you folks can communicate with me. All right. So, all right. Let me tell you a little bit about exams. Okay. Okay. Exams in this course are going to be obviously online. Yes, Matt, the website is Classmarker. I'm going to be sending information out through email. So if you miss something, don't worry. I'm going to get it to you. Okay. So don't worry about that. Okay. So exams will be held online using the classmarker.com website. Okay. So what you're going to do, hold on one sec. What you're going to do over the next week is register for an account, and it is entirely free. You do not have to pay for this. So in the next week, I need you to register for a free account with classmarker.com. Folks, to do that, you are going to need a special code from me, and I will get that to you later this afternoon, okay? Okay, so folks, later today, after we're done with our class time, I'm going to send through email an Excel file. And that Excel file is going to have your hack ID number on it, and it's going to have a unique code that you will use to register for the Classmarker website. So Pete, do we start this next Thursday, or is it due next Thursday? I'm going to ask you to set up your free account sometime this week, so to have it accomplished by next Tuesday at the latest, okay? You will receive an Excel file through email containing your hack ID numbers and codes next to them, okay? So when I send you this Excel file, I want you to open it up, look for your hack ID number, and use the code that is across from your hack ID number to create your free account on classmarker.com. Okay? All right. So I'm going to reiterate much of these instructions through the email that I will send this afternoon. But what I would like you to do by next Tuesday is create a free account on classmarker.com and take a practice exam that's already been set up. Okay. Okay. The practice exam does not count toward your grade, and it won't hurt you if you do bad, and it won't help you if you do good. In fact, the practice exam is simply to allow you to get comfortable with the website so that you're not confused about what's happening when you take the real exam in a couple weeks. Okay? So, any questions so far? Okay. So as I said, I'll be sending more information about that this afternoon in this afternoon's email. And so that should allow you to accomplish the goal of creating an account on ClassMarker for free. And let me just tell you about exams in general, okay? If we were together on campus, face to face, what I would share with you is that typically my exams have two components. Okay, there is a vocab component and an essay component, where one part of the exam I test your knowledge of vocab in the course. And in a second different part of the exam, I test your understanding of bigger topics or physiological topics, you know, such as how does osmosis work, that kind of thing. So obviously, since we're not together, I'm still going to do this testing with you, but all of it's going to be online, okay? So for every lecture exam, you will actually have 2 online exams to take. One is the vocab exam, and the other is the essay exam. Okay, so a week from this Thursday, when you take your first lecture exam, you will log into ClassMarker and you will see that there are 2 exams waiting for you. One will say it's a vocab exam, and you have to take that. Once you finish that, then you have to take the other exam, which is the essay exam. And you have to finish both of them on the same day, okay? So, the vocab exam is timed, okay? The vocab exam is timed, and you receive about 1 minute per question to take the exam. Okay. So for example, I mean, sometimes folks get a little nervous when they hear it's 1 minute per question. Please don't let that scare you, because one minute is actually a great deal of time. For example, if I asked you, let's see, what have we covered so far? Okay, certainly if I asked you what term represents the number of protons in the nucleus of an atom, well, we've already covered that, right? That's the atomic number. So recalling that from memory took about 5-10 seconds, and writing it out on your keyboard on the computer should only take another few seconds. So 1 minute per question is plenty of time to do the vocab exam if you've done some preparation and study and organize yourself, okay? The essay exam is also timed, and typically I give about 1 hour and 15 minutes to complete the essay exam. For the essay exam, you will have multiple questions that you have to answer. Typically 5, 6, 7 essays, and you have to answer them in that 1 hour and 15 minutes. Okay. All right. If you are in the accommodation program with Vicki Van Heist, then you will automatically be given double that time for your testing accommodations. Okay. All right. So, what are the nature of the questions for the vocab exam? You will have multiple choice questions and fill in the blank questions. Okay, that's the kind of questions you'll get to test your understanding of vocab. And for the essays, they will be more open-ended questions, where again, you have to explain a topic such as explain osmosis or explain how skeletal muscle tissue contracts, that kind of thing. At any rate, don't be scared of it. You all do just fine because I'm going to help you through it, okay? So let me momentarily stop sharing and ask you, do you have any questions at this time about the exams? Okay, so I'll send that information out this afternoon, and it will allow you to start getting comfortable with the Classmarker website. Play around with it. You're all very technology savvy. I don't think you're going to have any trouble. But if you do, contact me, bring it into class, and we'll discuss it so everybody benefits from it, okay? Any questions before we continue? Okay, folks. Well, I hope you're doing well. Hope you had a nice weekend. We're going to jump right back into it. I have just a few comments to finish from Chapter 2. Okay, I'm seeing some questions through the chat window at this point. Alyssa, do they need to be completed at a certain time? For the exam, on the day of the exam, the exams will open up at 7 a.m. and they will close at midnight. So you have basically all day to take the exam, okay? But let me say, once you begin the exam, you cannot stop it and you cannot pause it. You must finish it as soon as you start it, you must finish it, okay? Pete asks, do I need a paragraph to write the essay or a few sentences? Well, Pete, it depends on what I'm asking. Okay, some questions are going to be different than what you think. Science class is different from English class. So there might be questions where the essay is label the anatomy of a skeleton. So obviously you don't need to write sentences for that kind of essay. So it really depends on what the question is whether you're going to write a basic English paragraph or whether you're going to do something that's science related in the essay. Okay, I will say that generally speaking, I don't want long, flowery answers. I want you to get to the point and give me the correct information, okay? Science writing is not the same thing as English literature writing. These are different skills. Any other questions? Okay, folks, let's, Pete, is there any other way to study besides the practice test? Pete, the practice test you're going to get on Classmarker is basically a little quiz to get you used to the website, and I'm not even going to ask you biology questions. I'm going to ask you trivia questions. So the practice exam on the website will not get you ready for your exam other than getting you comfortable with the website. To get ready for your exam, you want to study your notes, read your chapters, do the practice questions at the end of each chapter. That will get you ready for your exam. Anything else? Okay. All right, folks, let's finish chapter 2. I don't want to spend a lot of time on chapter 2 today, but maybe 15 minutes covering some basic ideas, and then we're going to hit chapter 3 really hard today. Here we go. Okay, so here we will finish chapter 2. And let me bring up the chat window here. Okay, so I have the chat window back up so you can get a hold of me if you need me, okay? All right, so I believe at the end of last class period, we made pretty good progress talking about things. We were talking about the important properties of water, and I believe we did talk about pH. The last thing I want to do is simply introduce you to the major molecules of life, which basically finishes out chapter 2, pages 31 through basically the end of the chapter, page 39, okay? Now, I'm not going to go into a lot of chemical detail because I know none of you are going to become chemists or scientists, but I just want to introduce some ideas to you, okay? Okay, so let's talk about it this way. The 4 categories of organic macromolecules that make up life. The 4 categories of organic macromolecules that make up life. Okay. So, folks, can any question coming out here? I'll put it into your notes, but if you can answer it in the chat window. What does the term organic mean? Does anybody know what that means? What does the term organic mean? If you know, please type into the chat window. Okay, Alyssa says naturally forming, life-oriented. Okay, those are reasonable talking points. And I would expect, you know, if you're having a conversation at the supermarket, the term organic might come in, and obviously you're buying produce and things that are natural and related to life. That's all true. Can anybody tell me what the chemical or biochemical definition of organic is? Okay, Nick, thank you very much. Organic to a biochemist, to a biologist, means carbon atoms are present. Okay. The molecules of life that we currently know about in our scientific universe, you know, in the realm of understanding that humanity understands, all life is based on the element carbon. Okay. And so we put into your notes for chapter 1 that all living things are made of proteins, lipids, nucleic acids, and carbohydrates. Well, folks, every one of those 4 categories I just listed is an organic macromolecule. And by organic, I mean they are made of carbon atoms connected in different arrangements to form the molecules. Okay? All right. So the types are carbohydrates, lipids, proteins, and nucleic acids. Okay? Everything in your body that is not made of water is chemically related to one of these 4 big categories. Everything. Okay? So if it's not water, it is somehow related to a carbohydrate, a lipid, a protein, or a nucleic acid. And so we're going to take each of these and talk a little bit about them. Okay. Now, carbohydrates are shown to you if you have your textbook, and I hope you've solved that problem so that you can look at your textbook during lecture. It's extremely important. Carbohydrates are introduced to you on page 31 of your textbook. Okay, and you know, if you look at figure 213, there's some ideas there. I'm not going to ask you about the chemistry of things in this course because, again, we don't anticipate that you're going to need that in your future career. But at any rate, you know, you are a human being and you eat food and you want to eat nutritious food that is healthy for you. So sometimes it's important to have some basic understanding of these different types of organic macromolecules. Okay, so within the category of carbohydrate, okay, within the category of carbohydrate, there are different kinds. There are different kinds of carbohydrates, okay? Different types exist. There are monosaccharides, and an example, okay, when you say monosaccharide, that's the same thing as saying simple sugar. A monosaccharide is a simple sugar. A simple sugar is as simple as a carbohydrate can actually be. Okay? And so an example of a simple sugar would in fact be glucose. Okay? That would be glucose. So, you know, if you've had a piece of candy today or something, certainly it had some glucose in it. You can see the chemical structure for glucose on figure, well, 214 and figure 215, depending on what you want to look at, okay? So I'll put that into your notes here, figure 215 and figure 214. Okay. And gang, do not worry about the chemical structures. I'm not going to ask you to draw them out on an exam. That would be kind of hard to do on the computer anyway. And I'm not going to make you memorize them, okay? But be aware that glucose that you see in figures 214 and 215 is considered a simple sugar, and that means glucose is a monosaccharide. That is the basic level of carbohydrate that can exist. Now, simply because it's simple doesn't mean it's not important, right? Because let's think about it. What kind of disease do many folks have where they deal with problems with their glucose level? What's the name of this disease? What disease is characterized by high blood sugar? Yeah, diabetes, you bet. Okay, so, you know, this has relevance for everyday life. You know, this is extremely important, and many people unfortunately do struggle with diabetes. Okay, so, okay, glucose levels in the bloodstream become abnormal when a person is diabetic. Okay, the presence of excess glucose triggers body-wide inflammatory processes that harm tissues and organs. Okay, so that little simple sugar on page 32 of your book, when it's high in your blood plasma in an uncontrolled, unregulated state, such as in people that suffer with diabetes, that actually triggers an inflammatory process that is harmful to your body. Okay, we will talk about diabetes in some more detail in a future chapter, okay? So, you know, we're just touching the surface of the iceberg at this point. Do you have any questions? Okay, so let's see here. We have monosaccharides. I should probably do it like this. Monosaccharides exist. Okay. We also have, you can have small groups of these monosaccharides linked together, and that would build something that's generically called an oligosaccharide. For example, in figure 215, you see sucrose on the right side of the chemical reaction arrow. Sucrose is a disaccharide, but a disaccharide is just one type of oligosaccharide. Okay. Oligosaccharides are small chains of monosaccharides linked together. Okay. So as an example, figure 215 has sucrose. Okay. sucrose is a disaccharide, which is a type of oligosaccharide. Okay, so I'm sure you've probably heard of glucose. I'm sure you've probably heard of sucrose, okay? And that's what they kind of look at from their chemical perspectives. Now, we also have long chains of monosaccharides, and these are known as polysaccharides. Okay, polysaccharides would include starches and fiber. Okay, starches and fiber. Okay, so starches are important in your diet, and they have to be considered important, particularly for people that struggle with diabetes, because starches can in fact be broken down into simple sugars, which raise the blood sugar level. Okay, so starches are digestible by your body, and can be broken down into monosaccharides like glucose, which can elevate your blood sugar. Okay. All right. So, all right. So, question for you. What is the primary storage polysaccharide in plant tissue called? Pete, who came up with these long vocab words? Well, I don't know. You know, putting an English name on a chemical structure is very intimidating, and it does get worse. I promise you, it gets worse. Okay, what is the primary storage polysaccharide in plant tissue? Okay, cellulose is a nice answer, Alyssa, but no, I see why you answered that. You answered that at the bottom right of page 32. It says, picture B, cellulose is a structural polysaccharide found in the cell walls of plants. But notice that that statement says cellulose is a structural polysaccharide. And my question is about a storage polysaccharide. So thank you. You were very close. Okay. Okay. All right. Good. Good. It is, in fact, Julia, you got it right. It is starch, right? Starch. Now, I want you to think about this. When's the last time you enjoyed some French fries, right? Potatoes. The primary storage polysaccharide in those French fries, the potatoes, is starch. And obviously, if you're a diabetic and you eat a lot of potato, you need to be very careful, because that starch, which is a polysaccharide, can be broken down into glucose, which can raise your blood sugar level, which is why you got to be very careful with your diet. Okay. All right. Another question. What is the primary? Pete, am I saying potatoes aren't healthy? Pete, I'm saying that any food in excess can be damaging to you, right? You do need a certain amount of nutrients every day, but the amount you need depends on your personal body and your health status. Certainly the amount of carbohydrate that a diabetic should take in is a lot less than someone without diabetes. Okay. What is the primary storage? Yes, moderation is key, folks. Moderation is key. And the moderation itself will vary depending on whether you're a diabetic or not. What is the primary storage polysaccharide in animal tissue called? Okay. When you have some potatoes and you eat them, and that those potatoes, the polysaccharides broken down into sugar, how is that excess sugar stored inside your tissues? What's it stored as? What's it called? Okay, Catherine, nicely done. It is in fact glycogen. You see that at the bottom left corner of page 32. Okay, very nicely done, folks. It is definitely not fat, Quinn. Fat is a lipid, it's not a carbohydrate. Okay. All right. Okay, folks, good stuff. Now, for those that answered fat to my question about the primary storage polysaccharide, please understand something here because things are always more complicated under the surface. The primary storage polysaccharide is glycogen. And folks that suffer from diabetes, their body is not working properly. And so a lot of times if they have high blood sugar, rather than storing the excess sugar as glycogen, the cells of the body do perform an alternative chemistry which converts the energy into fat, and then they accumulate fat in the tissue. So there is a link here for diabetic patients. Okay, but right now, you know, I'm bouncing back and forth a little bit, but I am trying to give you the information on a normal, healthy human body. Okay. All right, folks. So now, last thing, and then we're going to move on here because I'm spending too much time on this. Basically, what I like students to know at this level is that every category of organic macromolecule has a basic building block. In other words, there's a foundational unit, a foundational molecule that can be used to build everything else that is a different type of carbohydrate. The basic building block for the polysaccharide or the oligosaccharide is the monosaccharide. So that is the basic building block of all carbohydrates. The basic building block of carbohydrates is monosaccharide. Okay? And there's different kinds of monosaccharide. You will learn that as you read the book, right? Glucose is a monosaccharide. Fructose is a monosaccharide. Arabinose is a monosaccharide. Deoxyribose is a monosaccharide. There's many, many different kinds of monosaccharides. Okay, any questions, gang? Okay, we're going to move on to the second type of organic macromolecule. This is your lipid category. Okay, lipids are made of substances that, generally speaking, do not dissolve very well in water. In fact, that's one of the reasons they're called a lipid, okay? Lipids are made of materials that generally do not dissolve very well in water, okay? Now, there's different types of lipids. Let's get these down here. First, we have triglycerides. Okay. Now, triglycerides can be made of both fats and oils. Okay. Right. So think of, think of canola oil or any kind of oil you might cook with. That is a triglyceride-based oil. Okay. And triglycerides have the chemical structure you see on page 33, figure 217. Okay. Triglycerides are made of a glycerol molecule attached to 3 fatty acids. A glycerol molecule attached to 3 fatty acids. Folks, into the chat window, somebody tell me what color rectangle is around the fatty acids on page 33? What colored rectangle? Okay, all right. Okay, Nick, right, red or orange perhaps, okay? I just want to be sure, folks, that you're looking at your book and that you're looking at the right area of your book, okay? It is not yellow, Pete. Okay, it's not yellow. So look very carefully, Pete. The yellow rectangle is around glycerol, but the red or orangish rectangle around the fatty acids. Okay, so triglycerides are made of a single molecule of glycerol attached to 3 fatty acids. I'm not going to ask you to draw any of that stuff out for the course, so don't worry about it, but you should be able to recognize these things as you're reading through the content. Okay. All right. So we have triglycerides. So Pete, you say the reds don't look like a rectangle. Well, let me see. On picture A, as I look at that chain of carbon atoms, they go vertically down, and it seems to me that the box around it does have a rectangular appearance. Okay. So let me know if you're still having trouble with that, Pete. Okay. So triglycerides are a type of lipid. And then phospholipids. Phospholipids are another type of lipid. Okay. Phospholipids are used to make the membranes of cells. They're used to make the membranes of cells. And you see an example of them on figure 218. Okay, figure 218, page 34. Okay. Each phospholipid is made of a glycerol molecule attached to 2 fatty acids. Okay. There's actually some other things here, but I'm giving you the core of it at this moment. Made of glycerol plus 2 fatty acids. Okay, so you can see that chemically speaking, phospholipids and triglycerides are very similar. Okay. And then finally, we have a third category of lipid, which are your steroids. Okay, now everybody knows about the word steroids. You've had it in the context of, people, athletes potentially abusing steroids and things like this. But the reality of it is that your body does use molecules that are chemically classified as steroids. Okay, now steroids look a bit different from triglycerides and phospholipids, and they're typically, their structure is characterized by a ring structure. Notice how their structure contains rings. And so I'll draw your attention to figure 219 here. Figure 219, as an example, right? Cholesterol. Now, we think about cholesterol from a dietary perspective, and everybody says, oh, I got to watch my cholesterol levels are getting high. But please understand, folks, your body does need a homeostatic, normal, healthy amount of cholesterol. Okay? Many of our hormones that we think about, particularly our gender or sex-based hormones, such as estrogen, progesterone, testosterone, these are all steroids. So these hormones are steroids, and they are also lipids, right? So these are things you've probably grown up with, you've probably encountered at various moments in conversation or in health class in high school, and that's what we're talking about here, okay? So the examples would include the sex hormones, estrogen and estrogen and testosterone is shown in your book, okay, of figure 219. Okay. All right. So, gang, the basic building block here, okay, the basic building block for all lipids is the fatty acid. The fatty acid. Okay, the basic building block is the fatty acid. Okay. So, question. What do Okay, I see your question, Quinn. Hold on one second. All right, so Quinn asks, is the steroids that we put into our body for the wrong purpose man-made? Yes, Quinn, it is. Okay. Today, through genetic engineering, we have the ability to synthetically make many of these molecules in the lab in large quantities. Okay. You know, if you were an athlete that wanted to dope your body with excess testosterone in the 1950s, what would have been the case is that somebody would have taken testosterone from a farm animal, maybe a pig, maybe a cow, and purified it out of their tissue and given you pig testosterone or cow testosterone. But today, with our advances in genetics and biochemistry, we can manufacture human testosterone and other sources of molecules in large quantities. Okay. And Quinn asks, also, if it is a lipid, how does that turn to muscle? Well, I don't quite understand your question, Quinn. Lipids are molecules, and muscles are tissues made of cells. And of course, cells contain lipids, right? So if you consume lipid material, those building blocks from the lipid can be recycled in the body to manufacture the components that a muscle cell needs. Does that answer your question? Pete, did you say they put steroid in animals? No. I'm saying, Pete, that as humans, we are also animals, right? Your dog is an animal, the cat is an animal, and your little sister is an animal. We are all animals. And so all animals use steroids. So cows have their own version of testosterone. Pigs have their own version of testosterone. And so it is possible when you slaughter an animal that you could biochemically purify that molecule out of the animal's body and put it into a human. Okay. That's how diabetes used to be treated. Okay. Many years ago, in the early days of diabetic treatment, insulin was purified from farm animals because we had no way of making excess quantities of human insulin. Okay, anything else? Okay, so question for you. If I can remember what my question was. Oh, that's horrible. Okay. Folks, you got me off track. I can't remember my question. Okay, so I will come back to it if I remember. Okay. So, third category of organic macromolecule or proteins. Okay, proteins are made of the basic building block amino acids. And your book gives you an example of some basic amino acids on page 35 at the bottom, figure 220, figure 221. Okay. Now, even though that seems fairly simple, I mean, there's basically 20 naturally occurring amino acids that your body uses to manufacture proteins. But because these 20 different amino acids can be arranged in different sequences, you can generate a tremendous diversity of different proteins that do things in the body, okay? So just as a point of interest, okay, most enzymes in your body are in fact made of protein. So here's a question that I do remember. What do enzymes do? Do you know? What do enzymes do? What makes an enzyme an enzyme? Have you heard this word before? Okay, so Nick, okay, so Quinn, you say it speeds up reactions. Nick says it breaks down different molecules. Okay, both are pretty good, but I do like Quinn's a little bit better. Okay, because Nick, not all enzymes break things down. Some enzymes actually manufacture things. Okay, so what do enzymes do? Okay, Jennifer, they create a chemical reaction in the body. They don't. I wouldn't say they create it, but they do accelerate it. Okay, enzymes are molecules that accelerate chemical reactions. Okay, so enzymes are proteins that accelerate chemical reactions. Please understand. Please understand. Most enzymes are protein, but not all protein are enzymes. Okay, most enzymes are protein, but not all proteins are enzymes. Do you see the difference in that statement? Okay. All right. So, okay. All right. Any questions right now? Okay. So, your book does hint at the topic of enzymes on page 37, and I wanted to be sure that I at least mentioned it to you and got it in your notes. Okay, and then enzymes are sort of important, basic beginner knowledge kind of things that you should know about. Finally, we have the fourth category of organic macromolecule, which is the nucleic acids. Okay, there's a variety of types here, DNA and RNA. DNA and RNA are types of nucleic acids. As you know, DNA is short for deoxyribonucleic acid. I just remembered my question, folks. It's going to come back at you. Hold on. Okay. RNA is short for ribonucleic acid. Okay. So I'm going to go back up here. Okay. On your screen, I've highlighted basic building block for all lipids is a fatty acid. So my question for you is this. What do you expect to happen when you drop a fatty acid into water? What do you expect to happen when you drop a fatty acid into water? It sits on top. Okay, I like your perspective, but no. Oh, I see. You're saying that because they don't mix well. Okay, fair enough, but that's not what I'm driving at. Okay, all right. Folks, you are correct that it's not going to dissolve very well. You are correct about that, but that's not why I'm asking this. So, okay, let me do this. Okay, so here's a question, and I'm going to shade it. I'm going to shade it, or I'm going to make it orange, I guess. Now, guess what? I'm going to take this question, and I'm going to repeat it and modify it down here. What do you expect to happen when you drop ribonucleic acid into water, right? Here's ribonucleic acid. There's a common idea that I'm driving at. Because yes, you're right, a fatty acid is not going to dissolve, but ribonucleic acid will dissolve. So that's not what I'm driving at. What am I driving at here? Well, what does the phrase fatty acid and ribonucleic acid have in common? Acid. Okay. So now I ask you, okay, Emily, you say it will release water molecules through dehydration synthesis. Emily, you're closer than anybody else, but you're definitely not using the language correctly. Folks, from our conversation last time, what makes an acid an acid? If you drop a fatty acid or a ribonucleic acid into water, what do you expect to happen? Emily, what do you mean with H+? Okay, good, folks. Good, very, very, very good. That's what I'm driving at. Exactly, Quinn, Nick, Alyssa, good stuff, okay? Exactly. The fatty acid or the ribonucleic acid is going to release some hydrogen ions into the water. That will increase the hydrogen ion concentration, which will change the pH, Alyssa. Very nice, okay? Okay, so Alyssa, here's a question for you. Alyssa, if you release some hydrogen ions, is that going to make the water more or less acidic? Okay, good. More acidic, that's right. So Alyssa, this question is for you. If the acidity increases, what happens to the pH value? Does the pH value go up or down? Okay, good job. That's right. Therefore, pH value drops. Good. Okay, folks, well done. Okay. Now, folks, when we talk about DNA and RNA, I mean, you're starting to get into my wheelhouse, the things that I really love about biology. But unfortunately, we're not going to talk about a lot of genetics in this course. But obviously, when you talk about DNA, you're talking about the material of your genes, right? The material of your genes. Okay, genes are made of DNA. And in fact, simply because it's very contemporary, the coronavirus has a genetic core made of RNA. Okay, the coronavirus has a genetic core made of RNA. In fact, the coronavirus genes are RNA, are made of RNA. Okay, folks, do you have any questions? Okay, if you have no questions, then I'm going to start chapter 3, okay? I never get as far as I want, but we got to cover as much of chapter 3 as we can so that we can stay on pace. So folks, here we go, chapter 3. Okay, chapter 3 is cells. All right. Okay. Some opening comments, okay? Couple things, okay? There are two major types of cell-based life on our planet, okay? And they are prokaryotic cells. Okay, now if you want to know a lot more about this, you need to take a micro course, which I do teach. You should take it in the fall. Okay, but at any rate, an example would be bacteria. All right, bacteria are prokaryotes. And a specific example of a bacterium would be Escherichia coli, which lives in your large intestine. Okay, Escherichia coli lives in your large intestine. Okay, so you should never feel lonely because it's with you all the time. And it manufactures some of the vitamins that your body cannot manufacture for itself, such as vitamin K. Now, prokaryote, the term prokaryote refers to no nucleus. Okay, so if you know what a nucleus is, prokaryotes don't have it. Okay. Now, the other form of life on the planet is the eukaryotic cell, and there's many kinds of eukaryotic cells. That would include fungi, it would include protozoa, it would include plant cells, animal cells, right? So obviously the animal cell that we are most interested in our course is the human animal cell, or Homo sapien. Okay. Okay. So we're going to spend a little bit of time talking about some of the basic anatomy of a human cell, which you see in figure 3.2. I will put a few things down in your notes, but hey, your book does a much more comprehensive job than I'm going to take time to do, which is why it is assigned reading for you, so that you are familiar with these things. Okay, so first off, I'm going to show you a video called The Inner Life of a Cell, which can be seen on xvivo.net. Okay, so folks, this is the first time in the course I'm going to show you a video. I think everything's in good shape with the technology, so let's see how things go here. So bear with me, I'm going to change what you see on the screen.
Are the captions on?
The captions are not on.
Okay.
Okay. All right, so folks, at this time you should see a website. You should see a website called xvivo.com. They are a scientific animation company. This company designs scientifically accurate cartoons, videos, animations, illustrations. If you have enough money to pay them, they will generate a beautiful video for you. Okay, I'm going to show you a shortened version of the inner life of the cell. And this is as a way to get you thinking about the topic of chapter 3 and what our body and what our cells are all about. So here we go. Just have a look at this, and then we'll come back after the video is over. So here we go. So, strange video, right? It's like looking at a science fiction movie. You don't know what all that is about. And so today we're going to introduce you to some of the pieces that you were looking at in that video. And I would encourage you to watch the video again as you study through this chapter and see if you can identify certain components of a cell. That was the short version. I believe there's like a 7-minute version of the same video on YouTube if you want to see the longer version. Okay? So, do you have any questions before we continue? Okay. Here we go. The video is about 7 minutes on YouTube, Pete. Okay. All right, chat window's back up. All right, so anatomy of a human cell, which is to say a eukaryotic cell. Okay, that's what we're looking at here. Okay. So, I'm not going to list everything out on page 44, but there are a few things that I do want to highlight for you. Okay. And so let's start with plasma membrane. Okay. The plasma membrane is the boundary of a cell. Okay. It defines the exterior from the interior. Okay. The plasma membrane regulates what kind of cargo can enter the cell and what kind of cargo can leave the cell. So it's involved in regulating the transport of material into and out of the cell. Helps regulate what material enters and exits a cell. And as I alluded to previously, and I think you know at this point, is that the plasma membrane is made of a phospholipid bilayer. Okay, so there's 2 layers of phospholipids with other molecules embedded in the bilayer. Okay, if you look very carefully on page 47 of your book, figure 3.6, right, you will see this magnified version of the plasma membrane that I'm talking about. Okay, so we'll get to all these things along our journey together today. Pete, you asked how long the cells are. Pete, that depends on what kind of cell you're talking about. An average epithelial cell in your mouth might be about 10,000 nanometers long, but an axon from a single neuron cell running from your hip down to your big toe might be over 3 feet long. Okay, so that's the interesting and complicated thing about cells, is that cells can have tremendously varying characteristics. Okay, but that has to be that way because cells need to do different jobs in the body. Yes, right, Quinn. If we're talking about a giraffe, you bet. Alyssa, is the plasma membrane considered part of the inside or the outside of the cell? Both. Okay, that's like asking, is the wall of your home inside or outside? Well, you appreciate there's an interior surface to your wall that's inside, but there's an exterior surface to your wall that's outside. The plasma membrane is the same way. It demarcates the inside from the outside. Okay, so it's part of both. Okay, so there's your plasma membrane. Number 2, cytoplasm. Folks, the cytoplasm refers to all the liquidy material okay, liquidy is a technical word. I am joking, of course. Refers to all the liquidy material and dissolved solutes in between the plasma membrane and the membrane of the nucleus. Okay, so material that's inside the nucleus of the cell is not part of the cytoplasm. All the material between the nuclear membrane and the plasma membrane is basically the cytoplasm. Okay, so obviously there is an abundance of water, but it's not just plain water, it's water with a bunch of solutes dissolved in it. Sugars, hormones, amino acids, fatty acids, all kinds of material floating around in the cytoplasm. Okay, so even though there's a tremendous amount of water that makes up the solvent of the cytoplasm, that water does not behave like water in a glass jar in front of you that you might drink. The water is very viscous, kind of like molasses or honey. So if you were to puncture a cell with a very fine needle, the water would not rush out. It would slowly ooze out as though you had molasses leaking out of a package, okay? Has the viscosity of honey. All right. Number 3, mitochondria. Okay, so that's plural. And mitochondrion is singular, right? Well, you probably, if you've had any other bio courses, you know that the mitochondria is famous for energy production. Okay, so mitochondria is heavily involved with the production of ATP. Okay, so how many mitochondria might be in a single leg muscle cell? How many mitochondria might be in a single leg muscle cell? What do you think, folks? Okay, Pete, thank you for answering, because that gives me a ballpark of what we're thinking about here. No, that's significantly too low. Okay, so Quinn says, all right, thousands. Matt, you're at 1,000, right? Okay, obviously it is going to vary depending on the size of the muscle cell and whose body we're talking about. But on average, you should think about 5,000 to 6,000 mitochondria in a single leg muscle cell. Okay, and that's just to give you an idea of how many of these little energy producing power plants we have in a typical muscle cell. Okay, now in an epithelial cell lining your mouth that doesn't have to contract or generate force for anything, then 50 might be a perfectly reasonable number of mitochondria for that kind of cell. Okay, so let's take a look at ribosomes. Okay, everything I'm hinting at is at least somewhat graphically cartooned on page 44 and throughout the reading of chapter 3 that you have to do in the days ahead. Ribosomes are nanomachines that manufacture protein. That's their job. That's what they do. Okay. What is the process of making protein called? What is the process of making protein called? What is the technical name that scientists have given to this process? Okay, to keep us moving along, it's called translation. Okay, translation is the term for manufacturing protein. Okay, gang. Feel free to throw out some questions there into the chat window. I'm going to keep rolling along here with notes. Next, we have the endoplasmic reticulum. Okay, this is a network of lipid-based membranes in the interior of a cell. Now, there's two types, okay? You have the rough endoplasmic reticulum, also known as the rough ER for short, and the smooth endoplasmic reticulum, okay? Now, the difference here, okay, the rough ER is called rough because on the surface of the membrane, you have a bunch of attached ribosomes. So the rough ER is called rough because of the presence of ribosomes. Okay. So the rough ER, obviously it has ribosomes and they make protein. So protein synthesis or translation can occur at the rough ER. In addition to this, in addition to protein synthesis, okay, in addition to protein synthesis, the rough ER is the site of phospholipid manufacturing. Okay, so in other words, the phospholipids that make up the plasma membrane of the cell, one of the primary sites that they are chemically put together is at the rough endoplasmic reticulum. Okay. Now the smooth ER, okay, it's called smooth due to the absence of ribosomes. So no translation, no protein synthesis occurs at the smooth endoplasmic reticulum. Okay, but what the smooth endoplasmic reticulum is involved with, okay, smooth endoplasmic reticulum is involved with the synthesis of fatty acids and also the detoxification of drugs in the body. For example, okay, for example, you know, let's say that you have a beer. Well, obviously you might feel a little tipsy after you've had a beer. Or if you've had 2 or 3 beers, you might feel a little tipsy, right? But the thing of it is that over time, you will not stay tipsy. You will not stay inebriated. Over time, you will sober up. And the reason you sober up is because there are enzymes, right? There are enzymes in the smooth endoplasmic reticulum that actually chemically modify the alcohol that you have consumed so you don't remain tipsy anymore, okay? After drinking alcohol, enzymes at the smooth ER will detoxify the alcohol so you sober up, okay? And particularly, the liver is a big deal here, right? The liver as an organ contains liver cells. The liver cells contain smooth endoplasmic reticulum. The liver cells in particular have a lot of smooth endoplasmic reticulum that's involved in detoxifying substances like alcohol or other drugs you might put into your body. I mean, think about it. If you have a headache, you might take an aspirin, but over 4 or 5 hours, the headache might come back because the aspirin starts to wear off. Well, why does the aspirin wear off? Because the aspirin is being chemically altered by your liver cells so that it can't do its job anymore of suppressing prostaglandin synthesis, which creates the headache. Okay? So the smooth ER is involved in detoxifying potentially dangerous substances in the body. Any questions? Okay. Okay, I hope everybody is still conscious. All right, very good. Let's continue here. Next part of a cell is the lysosome. Okay, the lysosome is the cell's recycling facility. Okay, I'm sure that probably you live in a town or municipality of some sort that probably once a week you put out some recycling materials, glass, plastic, cardboard, that gets taken away to a recycling facility to be broken down and reused to make new products. The lysosome is that facility for a cell. If a cell has damaged, worn out molecules or entire organelles like mitochondria, they can be recycled and the components of those materials can be reused to benefit the health of the cell. Okay. The lysosome can also be used for cell defense. Okay. Now think about it for a minute. Okay. If you think, maybe you live somewhere where you have a lot of property with some trees on, and maybe every once in a while a storm will knock down some branches of the trees, and they're a pain to get rid of. So maybe your folks or somebody you live with has at some moment in time purchased a wood chipper. So you can take a small branch of a tree, feed it through the wood chipper, and chop it up into mulch, right? You're recycling the wood for another purpose. Well, the lysosome can in a similar fashion be used as a wood chipper to recycle potential pathogens that are trying to infect the body. For example, if you have a virus or a bacterium that tries to infect the cell will attempt to run the virus through the lysosome as a wood chipper and destroy it. Now, obviously, it's not always successful because we still get sick from viruses. But the point is that your body will attempt to use the lysosome as a defense mechanism, as a defense structure. Okay, it can also be used for cell defense to recycle pathogens like a wood chipper recycles wood. Okay. Let's see here. I'm going to put two more, I'll put 3 more things into your notes and then we will move along. Okay, the cytoskeleton. Gang, the cytoskeleton is not a term that's present on page 44 of your book. But the cytoskeleton is a tubular network that includes things that are in your book, such as microfilament and microtubule. Okay, so I'm sure you can see those things on page 44. It's a tubular network that includes microfilaments and microtubules. And in the video I showed you, there was a moment in the video. Let me see here. Let me see if I can find it. Okay, right here. You see this funny looking odd thing that's attached to this blue sac, this blue bubble. and it's literally walking along this tube down here. Well, that tube is a piece of the cytoskeleton. That tube is a piece of the cytoskeleton. Okay, so what I'm trying to point out right now is the cytoskeleton is sort of a network of roads that are in the shape of the tubes. And machinery inside the cell will walk along these tubes to move cargo from point A to point B. Okay, can be used to move cargo around inside the cell. Folks, am I making sense? Does this make sense right now? Okay, thank you for the feedback. I need to know that you're live, gang. Yes, it is like a cellular highway. That's exactly it, Alyssa. Okay, so the cytoskeleton is like a cell highway. And if we're being honest, it also provides structural support for the plasma membrane so it doesn't collapse in on itself, right? So the cytoskeleton also has some structural responsibility as well. All right. So, right, all of these things we're talking about, let's be honest. I mean, chapter 3, the topic of chapter 3 is the cell. Well, you could take a semester-long course called cell biology, right? We could spend all semester on this topic. Chapter 2 is chemistry. Well, folks, we could spend an entire year on chemistry, right? So we really are just touching the surface of things. Okay, number 8 is the Golgi complex, okay, which is also known as the Golgi apparatus. Okay, that's probably an older term that I was taught years ago. Okay, but at any rate, what I'd like you to think about when you think about the Golgi complex is sort of equivalent to the cell's post office. Kind of like the cell's post office, okay? Think about it. You take a package to the post office. They might modify how you've packaged it for delivery. They might add some address information to it, whatever it might be. In other words, the Golgi complex is sort of the cell's post office because it helps the cell add biochemical zip codes to cargo so that the machinery of the cell knows where in the cell it's supposed to deliver that cargo. Okay? So the Golgi will modify cargo by attaching biochemical zip codes, right? And I'm using zip codes very liberally here. Biochemical zip codes to the cargo. So machinery can tell where the cargo should be delivered inside the cell. I mean, that's a fairly simple and neat idea to understand, but obviously the details are very complicated because it's all biochemistry. Okay, and the last thing I'm going to mention to you is the nucleus. Okay, I haven't mentioned everything, and I'm not going to mention everything, but the nucleus should be in your notes. The nucleus is a membrane-bound compartment where your DNA is stored and decoded. Okay, a membrane-bound compartment where your DNA is stored and decoded. Question: how many chromosomes are in a single human cell, right? The chromosomes are the biological structures that are made of DNA. So how many chromosomes are in a single human cell? 42. I hope not. 28, no. Okay. Nick, no. Oh, Nick, you could be right, but you need to contextualize your answer. Okay. So, okay, if we're talking about a diploid cell, which is what most of our cells are, like the cells of your heart are diploid cells. That means they have 2 copies of every chromosome. They got a copy from mom and they got a copy from dad. There are 46 individual chromosomes or 23 pairs, which is what you might have been thinking about, Nick. Okay, 23 pairs or 46 individual chromosomes. Okay. If we're talking about a sperm cell or an unfertilized sex cell, that is a haploid cell. There will be 23 individual chromosomes. Okay? Another question. Let's see here. I think I forgot a piece of information further up in your notes that I want to give you. So bear with me. I'm going to go back up to another location. Okay. Let's see here. What I forgot to give you for nucleic acids is that their basic building block is nucleotide. Okay. Nucleic acids, which include DNA and RNA, have a basic building block, which is nucleotides. All right. So that'll be complete when you get the notes this afternoon. So here's my question. How many nucleotides are present in a diploid cell? How many nucleotides are present in a diploid cell? Folks, what I am asking you is this. How big or how long is your genetic blueprint? How many nucleotides are present in a diploid cell? What I'm asking you folks is very similar to this. I could ask you this. How many letters are in all the words of all the sentences, of all the paragraphs, of all the chapters of your textbook? How many letters are in everything in your textbook? Well, obviously there's millions and millions, right? I mean, I don't know. Okay, Matt, so you say billions. Okay, Matt, you are on the correct scale. So the answer is about 6.4 billion nucleotide pairs of DNA. Okay. So what I'm saying is this is the size of the diploid human genome. There are 6.4 billion letters to your genetic blueprint that make you the person you are. Okay. When was the first draft of the human genome published? Do you know? When was the first draft of the human genome published? Oh, Lord. No, not the 1800s. Folks, how many of you, well, how many of you are younger than 19? Okay. All right. Folks, the first draft of the human genome was published in 2001. Okay. So, next question, then we're going to leave this topic. How much money in U.S. taxpayer funds did it take to acquire the first draft of the human genome? I guess everybody's safe talking about billions, right? All right. Yeah, it took, in 2001 dollars, it took just in excess of 3 billion US dollars. Okay, folks, $3 billion was what it took to get the first draft of the human genome. That is more money than it took to put a man on the moon. Okay? So we're talking about big, big, big science projects. All right, any questions? Okay, all right, folks, we got to keep moving here. So I'm going to shift gear now, and I'm going to talk to you about various processes that are capable of moving cargo across the plasma membrane of a cell. So this topic starts on page 48 and spills over for a couple pages here. Okay, 48, 49, and 50. Okay. Moving cargo through the plasma membrane. All right, so I suppose at this point I need to talk. We can break these processes down that move cargo through the plasma membrane into several categories. And so I'm going to break it down this way. There are passive processes in which the cell itself does not have to consume any biochemical energy to make it happen. And then there are active processes where the cell does have to spend biochemical energy to drive the motion of the cargo. Okay? So be very careful here. So passive means no cellular energy needed. Active processes means it uses cellular energy. And let's list out a few things first, okay? Under passive processes, we have things like simple diffusion. We have facilitated diffusion. And we have osmosis, and we're going to have to talk about these things. Under active processes, we have active transport. And then we have things that are involved with moving huge, huge cargo that would include exocytosis and endocytosis. And I may not have time to go into all of these things, but your book does talk about them, right? Anything I don't talk about but is in your textbook is fair game for exams. Okay? So let's talk about the passive processes. All right, simple diffusion. Okay, let me give you the big flowery language definitions of these things, and then we can talk about them. Simple diffusion is the movement of a solute from an area of high concentration to an area of lower concentration. Movement of a solute from an area of high concentration to an area of lower concentration. So, as an example, every time you drop a teaspoon of sugar into your coffee or tea to sweeten it, those sugar molecules dissolve in the water and they spread out. So where they entered the water is where the sugar grains were most concentrated, but as they dissolve, they begin to spread out through the volume of the liquid of your coffee or tea. That is an example of simple diffusion. Okay, so if you sweeten your coffee every time you go to Starbucks, you are employing simple diffusion. Notice that simple diffusion in my example can happen whether their cells are present or not, right? We don't expect or want any cells in our coffee. Simple diffusion is simply something that happens with a solute that's in water. That's what it is. Now, simple diffusion can be used to move cargo across the plasma membrane and get into the water or exit the water of a cell. It can do that, okay? But as I said, simple diffusion is a passive process. The cell will not consume any of its own energy. The cell will not consume any ATP to move the cargo. So, an important question. If a cell does not use energy to move cargo during simple diffusion, what is the source of energy to move the cargo? What's the source of energy to move the cargo? Because you see, an object will not move by itself unless something moves it. So, what's the source of energy moving the sugar in the cup of coffee? Okay, Quinn, nice. Gravity cannot be denied, right? If you put sugar at the top of your cup of coffee, gravity's going to start pulling it down. That's a good answer, and that is part of the response here. Okay? That's a good answer. I like that, because that tells you that physics is involved here. Okay? Not density so much, Matt. Density is a property. It's not an energy source. What I'm trying to get at, folks, is heat. Heat energy in the environment causes molecules to move around, okay? Heat energy in the environment is sufficient to move the sugar around in the coffee, right? Okay. Okay. So, simple diffusion. Do you have any questions about that? Now, the type of cargo that a cell can move through the plasma membrane using simple diffusion is going to be very tiny cargo, like oxygen molecules, O2 molecules, right? So oxygen is tiny enough that it can literally float in between the phospholipids that make up the plasma membrane and enter the cell by simple diffusion. So a cell can employ simple diffusion to allow oxygen to enter itself and be used, okay? So very tiny cargo like oxygen can be moved through the plasma membrane by simple diffusion. and tiny, well, let me clarify. Type of cargo, very tiny and electrically neutral cargo, okay, which oxygen fits the bill for. Okay, O2 is basically a neutral molecule because of the way the electrons move around both atoms. It's very tiny and the molecule is electrically neutral, so that is sufficient that it can go through the plasma membrane by simple diffusion. Okay. Questions? All right, so oxygen can enter the cells and carbon dioxide can leave the cells by simple diffusion. Emily, so how is the process of moving cargo through the plasma membrane different than moving the cargo through the cytoskeleton? Well, the cytoskeleton is a group of tubes. And so if you imagine the road outside your house as a tube and you walked along that road and you walked along that tube, that's how you move cargo along, right? That's how you drive your car along. But simple diffusion does not require a tube. It simply requires some water and very tiny cargo to fit through a confined or tiny space, okay? So what I am saying is this, Emily. Okay, hold on a second. I got to bring up a PowerPoint. Okay, so Emily, if we're talking about the plasma membrane of a cell, you're going to have these phospholipids that are like this. And there's going to be a bilayer of them. This will continue like this so that you have the membrane. And this would be inside the cell, and this would be outside the cell. What I am suggesting to you, Emily, is that for simple diffusion, oxygen molecules, okay, so O2, which I'll represent as a tiny little dot, okay, tiny little dots, these are oxygen molecules. These oxygen molecules are so tiny that they can literally squeeze in between the phospholipids and enter inside the cell. Does that make sense, Emily? Okay. So obviously very large cargo, such as a giant protein. Okay, so we might have a protein, which, you know, I can't draw a protein, but, you know, protein might have a very globular kind of weird shape to it. This thing is way, way too large to get through here. It'll never get through there. So you have to do other processes to get proteins across the plasma membrane. In terms of the cytoskeleton, right, again, all we're saying is for the cytoskeleton is that inside the cell, you have these little tubes that machinery can use to move cargo along. That's all we're saying. Okay? Other questions? Okay, let's continue here. All right, the second type of passive process is facilitated diffusion. Okay, this is the movement of a solute movement of a solute across a membrane with the help of a carrier protein from an area of high concentration to an area of low concentration. Okay, so notice how the definition changed, right? Across a membrane with the help of a carrier protein. You can see that on page 48, figure 3.8, okay? Figure 3.8 is showing you that. Anybody out there in the virtual world, what is the color of the carrier proteins in figure 3.8? Okay, thank you, Jennifer. Exactly, purple, okay? Okay. So folks, these purple proteins, I mean, obviously they're not really purple in real life, but they're drawn purple to make your textbook interesting. These proteins are the carriers that help cargo move across the plasma membrane. Okay, so the type of cargo here can be large proteins, large and electrically charged cargo. So as an example here, a protein, now let me do something a little simpler. Glucose. Glucose is a fairly large molecule compared to oxygen, right? If you remember, the molecular formula of glucose is C6H12O6, right? So glucose has 24 atoms, right? 12 and 6 is 18, 18 and 6 is 24. So there are 24 atoms in glucose, but breathable oxygen only has 2 atoms of oxygen. So that means glucose is a physically larger molecule, okay? And so you need special carrier proteins to safely get these large molecules through the plasma membrane. Okay, Pete, you asked, why is it always purple or red or blue? I think that's just the coloring conventions of illustrators and artists that draw these things, Pete. Okay. So, so an example would be glucose. A second example would be a hydrogen ion, H+, right? A hydrogen ion. A hydrogen ion is extremely tiny, but it is electrically charged. So it also needs to get through the membrane with the help of a carrier protein. Okay? All right. Any questions at this time? Now, I mean, these are some interesting things. And honestly, even if we were in class together, we would not do any experiments in lab that illustrate this. But we do some osmosis experiments with our Biology 101 students, our undergraduate biology majors. So if you're interested in that kind of thing, you might want to take that course because you get to do some kind of fun experiments in lab when we actually are able to physically be on campus. Okay. All right. So the third thing that I'll talk to you about, the third passive process is osmosis. Okay. Now, osmosis is a very involved topic. Without a doubt, it's probably one of the more complicated things we talk about here in this chapter. So osmosis, okay, let me give you the big English definition. This refers to the movement of water across the membrane from an area of high free water concentration to an area of low free water concentration. Okay? This refers to the movement of water across the membrane from an area of high free water concentration to an area of low free water concentration. The key to understanding osmosis is definitely understanding what this concept of free water is, or appreciating that term free. Okay. So let me go back and ask a few questions here. Okay, let me go back and ask a few questions. So I'm going to stop sharing. Okay. Okay, so okay, so again, folks, it's very strongly encouraged if you would please allow your video cameras to be on. It makes for a better class. Okay. At this point, let me ask. Okay, Julia, are you online, Julia?
Yes.
Okay, Julia. Go ahead and keep your microphone unmuted. I want to ask you a couple questions around osmosis, okay?
Okay.
Okay, Julia. Julia, are you married?
No.
Okay, so in my funny little story here, we're going to have you be married, okay?
Okay.
So Julia, let's say that you are home and you are starting to prepare a dinner for your husband and some friends that you want to entertain that evening to have a little dinner party. Okay?
Okay.
And let's say that you ask your husband to bring home a head of lettuce so that you can prepare a nice salad for the dinner party. Let's say that when your husband gets home, he hands you the lettuce, and you look at him with that look of disgust that happens when a husband brings home a wilted head of lettuce. Are you with me?
Uh-huh.
Okay. So your husband has brought home a wilted head of lettuce, and neither of you have any time to go back to the store and get a better head of lettuce, and you do not have the option of throwing the lettuce out and simply cooking something else. You need to prepare a salad. What could you do as a cook, as a chef, to improve the condition of the wilted head of lettuce? By wilted, I mean it is all dehydrated and shriveled up. So what could you do as a chef to make the wilted head of lettuce be better for your guests?
Maybe run it under cold water.
Okay. Well, you're starting to get at it. The water doesn't have to be cold. The coldness is simply your preference as a human being. You prefer to eat chilled salad as to warm salad. That's simply a taste preference. So the water temperature doesn't matter. But okay, you could put it under some cold water. And I would suggest to you as a cook or as a chef, you should go even further and actually soak the head of lettuce in a bowl of water. In other words, you should get a bowl of water, put the lettuce in it, and let it sit for a while. Have you Have you done this as someone cooking food before?
No.
Okay, but have you seen it on any cooking shows?
No.
Okay, do you cook any food?
Not really, just like eggs and pasta.
Okay, do you eat any food?
Yes.
Okay, there, I got you. Okay, you eat food. Okay. So the point of it is that, you want to get some water back into the cells of the lettuce so that they plump up and so the head of lettuce is not so wilted and dehydrated anymore. So I'm almost done picking on you. Hold on. Julia, the water that you use to put the lettuce into it, should it be hypertonic, hypotonic, or isotonic?
Hypotonic.
Could you spell that for me, please?
Hypotonic.
Folks, Julia says that you should place the lettuce in hypotonic water. In the chat window, type agree or not agree. All right, it looks like everybody's agreeing, so either you all understand it or some of you are going along for the ride because you figure it's safest to do that, right? Okay, so Julia, hypotonic is correct. Where are you going to get this hypotonic water?
From the tap.
Okay, nicely done. In other words, the tap water that you take out of your kitchen sink, out of any faucet at your home, is always hypotonic to the content of the cells, which is why water is able to go across the plasma membrane of the cells making up the lettuce, and the cells of the lettuce can rehydrate and inflate back up. Okay? So nicely done. So Julia, you did a good job, okay?
Okay.
All right, so let's take this idea that we've been talking about and put some terminology down and understand what's happening here. Okay, actually, let me show you a little video on YouTube about osmosis. The video I'm going to show you starts out talking about simple diffusion, and then it transitions into the topic of osmosis. Okay, so let's watch this, and I think it will help make this talking point more understandable for you. Okay, so folks, what I'm getting at here is you may have learned about osmosis in the past, but what I find is that many times students learn a trick to answer a question right, but it doesn't mean you actually understand how it works. And we're trying to break that down so you understand. Okay, so bear with me. Here we go for the cartoon. Okay, so on YouTube, I'm going to this video. I got it by searching how osmosis works, and it's called Osmosis High Definition Animation by McGraw-Hill. Here we go.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Okay, folks, good stuff. So let's continue our talking about osmosis. So some comments. Okay, sorry about that. Okay, so remember when you're talking about osmosis, well, let me ask you this. When we were talking about simple diffusion and facilitated diffusion, were we talking about the solute or the solvent? Okay, once again, when we were talking about simple diffusion and facilitated diffusion, were we talking about the movement of the solute or the movement of the solvent? That's correct, Matt, solute. But please understand, for osmosis, we're interested in talking about the solvent, not the solute. Okay? Notice how osmosis involves the motion of the solvent, not the solute. Okay? For the first two items we talked about, simple diffusion, facilitated diffusion, we were talking about oxygen or sugar or something moving, that was the solute. But in osmosis, I'm talking about the water, which is the solvent. Okay, so this term, question here, what does the term free mean in this conversation? Okay, what is free water? What is free water, folks? Okay, all right, thank you for your participation, folks. Please encourage you all to participate. It's okay to be wrong. I just want you to participate. Okay, so Gwen, you said something there that caught my attention. The water isn't attached to a molecule. That's exactly it. Free water means the water molecules are not interacting with another molecule. They're not attached in any chemical or electrically attracted way to another molecule. This is when water is not interacting with other solute molecules, okay? This is when water is not interacting with other solute molecules, okay? If a solute can interact with water, then the free water concentration in that area goes down. Okay, if a solute can interact with water, then the free water concentration in that area goes down. Okay, heavy water is due to isotopes. That doesn't have anything with the context of what we're talking about now. Okay, heavy water would work the same way as normal regular water. Okay, so let me go back to PowerPoint here. Okay. Okay. So let's try this, folks. Let's say we have the following scenario. Okay. Let's say that I have, oops, one moment. Let's say that I have, oh, doggone it. Sorry, folks, having some tech issues here. Okay, let's say I have a jar of water, okay? In this jar, I have a bunch of water, okay? And let's say that I scraped the inside of your mouth with a toothpick, and I scraped off one of your cells, and I put your cell in this water. Obviously, this is not drawn to scale because if it was, you would never see it. Okay, so we have one of your cells here. All right, and in your cell, which I'm going to momentarily make bigger. Now, see if this helps at all. Okay, so you have a cell in the water, and let's say that in your cell, I have 6 water molecules. Okay, so I have H2O, 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 and H2O. I have 6 water molecules. And then let's say, through the magic of technology and entertainment purposes, let's say that I take and through the magic of 3D printing, I print a box that's the same physical dimensions, well, not the same physical dimensions, but the same volume as the cell. Okay? So, in other words, I have 6 water molecules inside the cell, and I also have 6 water molecules inside the box. Actually, I'm going to stay with 4 at this point. Okay, I'm going to stay with 4 inside the box. Okay. So, all right. I'm trying to ask you the question, what are the number of free water molecules? Okay, and we'll divide this between box and cell. Okay, so type into the chat window at this moment in our conversation in the drawing, how many free water molecules are in the box? 4, you got it. Okay, so I got 4 free waters in the box. How many free waters do I have in the cell? Okay, good job, everybody. Got 6 free waters there. Very good. Now let's say I take a salt shaker from your kitchen table. Okay, let's take a salt shaker and let's see, what's a good color here? I guess orange maybe. let's say, take a salt shaker, which obviously has sodium chloride in it. And let's say I drop 1 molecule of sodium chloride into the box. Okay, I'm going to drop that into the box. Okay, now, obviously in water, sodium chloride is going to separate into sodium ions and chloride ions. So that means that the sodium ion might be able to interact with this bottom water, and the chloride ion might be able to interact with that top water molecule. Okay, so now what's the number of free water? What's the number of free water with the salt present? Okay, so in the box, how many free waters do I have? Okay, you guys got it. All right, I now have 2. And how many free waters do I have in the cell? I still have 6. That's exactly right. Okay, so let's think about our osmosis definition. The movement of water across the membrane from an area of high free water to an area of lower free water. So the question I'm proposing to you is water going to leave the cell and enter the box, or is water going to leave the box and enter the cell? Which way is the water going to go? Okay, Nick says it's going to leave the cell. How about the rest of you? Okay, you say it's going to go to the left, Emily, so that means, it's going to leave the cell. Okay, that's right. So in our hypothetical scenario, the water is going to leave the cell and enter the box, because the purpose and goal of osmosis is to get the same number of free water molecules equal on both sides of the membrane, or on both sides of the imbalance, if you will. Okay, so water will leave the cell and enter the box. Okay. All right, folks. Do you have any questions right now? All right, let me go back to the, so one more question here. One more question for you. So this area that I'm highlighting with the yellow marker, the box with the chloride in it and the sodium in it and the water, okay, this liquid in here, is it hypertonic, hypotonic, or isotonic relative to the water inside the cell? Is the water in the box with the salt hypertonic, hypotonic, or isotonic? Okay, is the water hypertonic, hypotonic, or isotonic? Okay. Thank you, Pete, Emily, Matt. Okay, very good. All right, that's what I was hoping you would see, that in our example here, this water is in fact hypertonic. Okay, so we need to define that and put it into your notes. Bear with me, folks. I might need about 5 minutes past our time together, okay? Bear with me here. All right, so let's make sure that you're okay here. Some terminology. Hypertonic means that there's more solute on one side of the membrane than the other. Hypotonic means there's less solute on one side of the membrane than the other. And isotonic means there is equal solute on both sides of the membrane. Okay. All right, folks. Do you have any questions about osmosis? All right. Okay. Well, okay. So do you have any questions about any content we've covered today? Okay, so I'm going to stop sharing my screen, okay, and simply remind you of a few things, okay? Later this afternoon, I will be sending you that email with the registration information for the Class Marker website. Please create a free account. Remember to write your login credentials down, keep them safe, keep them private, because you will need them the rest of the summer. And then please take the practice quiz online in the next few days. It's only 5 questions. It will not take long. Next, we have lab again on Thursday. The lab on Thursday is about the microscope. Right? There's no way that we can do things when we can't touch a microscope, right? But again, we will walk through the content of the activity. So please, for Thursday's lab, read through the lab manual for exercise 2 and complete as much of the activity that you can without actually physically touching a microscope. You can do some of that. For example, one of the things that you have to do is label the parts of a microscope for activity number 2. Right? For activity number 2, we want you to label the parts of a microscope. To do that, you read the information on page 14 of your lab manual. Okay? So what I'm saying is there are aspects of the lab activity for Thursday that you in fact can do simply by reading it. So do as much of that content as you can for Thursday's lab. That way when we walk through it, we don't have to spin our tires on things that you can do on your own. And we will talk about whether you got your answers correct or not. Okay? So, typically in this course, we teach the microscope and how to use it, because the next lab, we would have you use the microscope to look at the tissues, to learn about epithelial tissue, muscle tissue, connective tissue, and nervous tissue. So it's going to be a little rough around the edges, but we will do our best to accomplish those goals. Okay? So please sign up for Class Marker. Please do as much of exercise 2 in your lab manual as you can for Thursday's class. Do you have any questions?
Yeah. What page in the lab manual?
Okay, well, the lab manual, exercise lab number 2 begins on page 13.
Okay.
So read through it and do as much as you can without actually being in front of a microscope.
So from page 13 to
page 13 till the end of it. Okay.
Okay.
It might be helpful to go to Google Images and type in microscope, and it'll bring up a microscope that you can look at while you're doing things. Okay? Do you have any other questions? Okay, stay focused. Be sure your study time is starting to get serious, gang, because a week from Thursday, you will have an exam. Okay? So I will see you on Thursday. Take care. And send me an email if you have any questions. Okay? See you later.
Professor John. Hello.
Yes, hi.
Hi, Professor. I have a quick question because I haven't been in class for a few years now. How could you advise me what's the easiest way to learn all these terminologies, like without mixing them up?
Okay, hold on one second. Fiona, I'm having a real hard time understanding you. Can you get closer to the microphone or maybe type your message into the chat window?
Okay.